is Olaf Chmura, and I would like to present results of our little research which looked at gut microbiota alterations in patients with COVID-19. General information about COVID-19. COVID-19 is a major pandemic infecting more than 22 million people worldwide. Human gut microbiota is a complex microbial community that influences human metabolism and immune function both in physiological and pathological states. While this SARS-CoV-2 is found in gastrointestinal tissues, little is known regarding the exact role of gut microbiota in SARS-CoV infection. The aim of this study is to establish whether the changes in the microbiota during the infection correlate with patients' outcomes. We analyzed data from 9 clinical studies. Here we can see our PICO. P is patients infected with SARS-CoV virus. I is analysis of microbiota. C, control group of patients with confirmed lack of COVID. And O, correlation between clinical characteristics and altered microbiota. Here you can see our Prisma graph. As you can see, there aren't many studies concerned with this topic. Here is the list of included studies. And here are our main findings. Current results are significant of altered microbiomes in patients with confirmed COVID-19 when compared to healthy controls. Those results are inconsistent across different samples. Most common findings include reduced bacterial diversity and lower relative abundance of beneficial symbionts and higher relative abundance of opportunistic pathogens. Only one study analyzed lung microbiota patients. It was significant of great presence of pathogenic bacterial strains. The downregulation FACE microbiota pathway may be a reason for diarrhea in SARS-CoV-2 infections. Some studies observed correlation between disease severity and abundance of Coprobacillus clostridium pathogens. What else? The detected changes persist throughout this hospitalization period and may be still present after the clearance of the disease. And according to one of the studies, a 5 gut microbiota biomarker is able to distinguish between COVID-19 and influenza A patients with area under curve of 0.89. Such analysis is also promising differentiating tool between those two diseases in the upcoming flu season. What's more is that detected changes to conclude that while the current data does not allow us to draw clear conclusions in regard to microbiota prognostic and therapeutic role, it serves as a solid foundation for future more widespread studies. We believe more studies should be initiated in order to fully explore the potential use of gut microbiota prognostic and therapeutic role. It serves as also the potential use of 16S ribosomal RNA sequencing as prognostic and diagnostic tool should be examined. Such analysis may also provide data necessary for development of microbiota-based personal therapies. That would be all. Thank you for your attention.